Ladies and gentlemen, can you take your seats, please? We need to start this afternoon's session. I'm going to introduce our speaker, keynote speaker this afternoon, Professor Jacco Aby. He works at Wageningen University, and he has a personal chair there, and his research is focused on the functional genomics and ecophysiology of food-related microorganisms, including starter and spoilage bacteria and foodborne pathogens. And you can see the title of his talk now. So welcome, Professor Aby.
Thank you, Janet, for a nice introduction. And happy to be here and to present this afternoon. And I want to thank the organizers for the invitation. So I actually fit very well in the additional subtitle of the conference, Next Generation Challenges in Food Microbiology, because I hope to present you some challenges. And also because I think there's indeed room and space and energy for new generation scientists that can actually pick up some of the current ideas that are running around. So today I will discuss diversity and substrate utilization capacity in Listeria monocytogenes, impact on transmission and violence. And while thinking about this presentation, I realized it would be very broad and decided to focus on one of the favorite topics we are dealing with at the moment in a group, and that is bacterial microcompartments. Bacterial microcompartments are actually organelles and prokaryotes in bacteria, which have gained a lot of attention in certain research areas in nanotechnology and biotechnology. There's also an interest in foodborne pathogens, especially E. coli and Salmonella. But there's a lot of studies and approaches used in bioinformatics, whole genome comparisons, and there's very little information actually about physiology. So I was very much intrigued by these protein organelles, and so there's organelles like in eukaryotic organisms, but these organelles are not enclosed by membranes, but by proteins. So this is really unique. And I hope to share my enthusiasm and inspire you for further analysis and thinking about probably maybe future research in this area. So transmission in the food chain is supported by substrate utilization, both in aerobic and anaerobic conditions. And in this presentation, I want to zoom in on anaerobic conditions because that's where the microcompartments, these special organelles, come into view. And they actually facilitate metabolism of non-fermentable substrates. They actually allow accumulation and condensation of certain enzymes into the compartment, so it makes the metabolism more efficient. And as I will show you later on, there's another reason, and that's because there's volatile intermediates that are toxic. So there's several reasons why the microcompartments are essential for supporting metabolism of certain compounds and anaerobic conditions. And I will show that these systems are actually activated in Listeria monocytogenes under certain conditions, despite the multiple level control. And I will also give some examples about the growth performance and anaerobic conditions, and actually link this to conditions that they may encounter in modified atmosphere or vacuum-packed chilled stored foods like cold smoked salmon. So we see a happy guy here because he defended his thesis in December. He actually did his PhD on these microcompartments, addressing several topics that are interesting. And he actually managed to submit 7 papers. So 5 of them you can find in online journals, and 2 of them are in bioarchives. And of course, if you're interested after this presentation, you might contact me for a PDF of the thesis or a hard copy. So this is Andy, and he did a great job. And we actually picked up, starting with a cartoon of Joe Kervel's lab in Berkeley, that shows these compartments in a bacterial cell. And as I mentioned, they contain metabolic enzymes enclosed by a protein shell that actually allow metabolism of compounds that actually have volatile toxic intermediates. So in this way, they are separated from the cytosol, and bacteria can actually consume and metabolize these substrates and actually produce energy that allows them to grow under these stressful conditions. And the basic composition is given in the middle. So there we have this enclosed microcompartment. There's a wide variety of proteins involved in the shell. That's also why I has drawn the interest of biotechnologists and nanotechnologists, because they actually like to play around with proteins and then create novel nanoreactors. So what happens is that the substrate can be taken up via specific diffusion proteins. Inside, the substrate is converted by a couple of enzymes. The volatile intermediate is retained during the, let's say, the existence of the compound and before it is transferred to the next compound. And then at the end, the intermediate, because there's further metabolism in the cytosol, actually will proceed. And in the cytosol, the ATP will be generated. So this shows one of the papers of the Kerfeld group, showing the diversity in bacterial microcompartment clusters. And what you actually see is that the largest cluster that has diversity in these compartments is actually the firmicutes. Well, Listeria is in this group. And the representatives are predicted to include over 20 distinct type of microcompartments. That doesn't mean that it's 20 different substrates, but it points to a wide diversity and the type of clusters and the type of enzymes and the type of shell proteins that are involved in the different species. And as mentioned in several talks this morning, also in the keynote by Luca, and so you can actually look at the diversity and try to translate it to functionality. But the biggest part of these microbes have not been cultured. So there's still an enormous reservoir of information we have, and that has to be applied to what are they actually doing and what is the role. So in the analysis, it turns out that Listeria contains membrane compartment dependent utilization systems for ethanolamine and propanediol. And these clusters are mainly present in human pathogens, among other Salmonella, and only very limited and beneficial bacteria. Up to recently, the only studied bacteria that actually produces microcompartments is Lactobacillus reuteri. And we recently provided evidence and also have shown that Propionibacterium freudenreichi, a well-known probiotic and also starter bacteria, also produces these microcompartments and can actually metabolize propanediol. So this is the overall design. On the left, I gave the structures of ethanolamine and propanediol just to visualize it. They're very simple compounds, C2 and C3 carbon. And normally they cannot be metabolized without oxygen. So you need these special features and combinations of characteristics of the cell to be able to metabolize these compounds and generate energy. So if you look to the right, we can actually see the catabolic so-called metabolisome, because they actually metabolize substrates. So the substrate, it could be ethanolamine or propanediol, is going in. There's a signature enzyme that actually produces the aldehyde, and then the aldehyde is rapidly converted into alcohol by alcohol dehydrogenase. And to maintain redox balance, another substrate that is converted to an aldehyde is actually going the other way. That's via aldehyde dehydrogenase and a phosphotransacylase. And this produces SL phosphate on the bottom of the carboxysome microcompartment. And then the phosphorylated compound moves out, and this is a high energy phosphate bound. And actually ATP can be generated by SL kinase. So what happens is that you have a unique situation where you have non-fermentable substrates. They can actually be metabolized in this very efficient way by combining and concentrating all enzymes. And actually the energized compound, the SL phosphate, moves into the cytoplasm and is actually converted to an acid, and ATP is formed. And at the basis you see the simple, let's say, construction of this, the gene cluster. But you have to realize that there are multiple shell proteins and there's multiple components that make up the enzyme. So these are also 2 or 4 component enzymes. So that brings me to Next slide. So these compounds, ethanolamine, and also glycerol, as you'll know in a moment, they are present in membranes, and all living forms have membranes. And both bacteria, plants, animals have large amounts of phosphatidyl ethanolamine in their membranes. So it's one of the main components in let's say, double-layered membranes. So it's present everywhere. It's present in food. It's actually present in the GI tract because we have a shed over of the epithelial layer, and then also lipids are released. And these lipids, actually indicated in the center, can be attacked by a number of enzymes, diacetyl esterases or phospholipases, phosphatases, and they can actually cut off the acyl chains, release glycerol, and that can also act as a substrate and release ethanolamine. And so that's actually indicated on top. Yeah? Glycerol can be converted to propanediol. So there we have the two substrates that can be actually utilized by Listeria putatively and in a pathway via the microcompartment. Propanediol is actually also produced from rhamnose metabolism. And rhamnose is one of the main carbohydrates, one of the main hexamers, and plant cell walls. So there's several levels where propanediol and ethanolamine can be produced or released in foods or in our intestine. So if you then look at the current status of the research, on top I indicated the several food products and also where you can actually encounter these compounds, so ethanolamine, more in lipid-rich foods, propanediol also because of glycerol conversions, dietary fiber, and that contains large amounts of rhamnose, so rhamnose metabolism could supply propanediol. And 1,2-propanediol is also a food additive, so it's also added to food. As far as I know, it's a sort of a stabilizer. So the current situation is that there's BMC-dependent propanediol utilization and ethanolamine utilization gene clusters identified. There's a role suggested in transmission in foods based on gene expression studies. And that was a very nice study from the group at Cornell already in 2015. So that actually showed transcriptome analysis of the adaptation of Listeria monocytogenes to growth on vacuum-packed cold smoked salmon. And what they actually showed was activation of expression of PDU utilization and also ethanolamine utilization genes. So they hypothesized that it could perhaps indicate that they were actually using, let's say, these compounds as a growth substrate. But this is just hypothesized based on gene expression. And together with what I mentioned about the microcompartment studies in biophysics and also nanotechnology, this paper really triggered me to think about a project where we could actually analyze and confirm the role of microcompartments in Listeria. There was another indication that they could play a role in violence that was described in a study by Chart et al. They actually constructed a mutant in the PTU cluster. And in mouse model studies, they actually could show that the wild type had a longer, let's say, perseverance time in the mouse GI tract. So actually, with inactivation of one of the enzymes in the pathway, the transition time was actually shortened. So it actually was shed earlier. And there were also some indications that maybe intracellular growth was affected, but that was not very clear. So these were some indications, but no proof of the pathway. So that brought us to three, let's say, approaches. First, we wanted to analyze the distribution of these type of clusters in Listeria species, because now there's more, let's say, Listeria species known. And it was interesting to see whether there would be any additional, let's say, distribution there. We also wanted to investigate the role of vitamin B12 or cobalamin. This is a cofactor. It has been suggested that some of the utilization pathways via microcompartments are B12 dependent, whereas others are not. And there were indications that the Salmonella B12 cluster was involved in this conversion. So we want to see whether this was the case also in Listeria. And we combined growth analysis, metabolomics, proteomics, transmission electron microscopy to investigate whether indeed growth was supported by activation and reproduction of microcompartments, and also, of course, we check metabolomics. And finally, we actually decided to investigate a possible role in violence using CACO2 model system, which is, of course, just one model system, one model in vitro system that you can use. But what we actually wanted to see is how do the cells behave if they are non-induced and if they are induced for the system. So we really knew that we have control cells that were not induced, and we had control cells that had produced these compartments and could metabolize these lipid derivatives. So that was actually the final part of the study. And for today, I will, of course, finalize with the discussion and a short outlook. So this is the B12 cobalamin cluster in Listeria. There's a very nice recent paper in Frontiers in Microbiology by Anas et al., so I advise you to read it if you're interested. And so this is the propanediol cluster, the ethanolamine cluster, and the vitamin B12 cluster. And why is this apparent, and why is this to a certain extent logic? Because the key enzyme, the signature enzyme involved in these pathways is actually B12. So it makes sense that if you activate these utilization clusters and want to actually activate and use a substrate, you also need a B12 cofactor, otherwise the signature enzyme doesn't work. So the assumption is that, okay, and the whole system is activated depending on the substrate, and so there's activation of PDU if you have propanediol, there's activation of ethanolamine if you have ethanolamine, but anyway, and you need activation of the B12 cluster. So that's a very interesting concept, because if you look at the vitamin B12, it's a very, very complex molecule. So it also indicated if you look at the genes, and this is not all the genes that are actually involved, you have to tune up a significant enzyme machinery to produce this compound. And on the other hand, if you look at all the other genes, you have to tune up a very significant machinery to produce all the shell proteins and compartments and also the enzymes. So, and I won't go into detail, but there's at least 8 regulatory levels of regulation. And so there's the sigma B, there's other sigma factors, there's at least 4 small RNAs, there's a 2-component system, there's a B12 riboswitch, and this indicating that the cell is really acting to have a fine-tuned control whether to activate or not the whole system or only parts of it. So that's why we took up actually another topic, and that's what I relate to this, that we wanted to see whether addition of B12 is actually needed to activate the system and support growth. And this was inspired by a paper in 2018 from University Groningen, where they actually characterized a B12 transporter from Lactobacillus delbrueckii. And it turned out that it had similar, probably similar transport system in Listeria, but it was of course not confirmed at that time. So there were several options to analyze regulation of these metabolic pathways. So just very quickly, Andy looked again for distributions and including novel Listeria species, and actually we still find it's only present in Listeria sensu stricto. And so that actually indicates it's not unique for monocytogenous, but this also fits in the analysis that it might support growth of Listeria species and a wide range of environments where these compounds are probably available or produced. So the first experiments Andy did was actually looking at we first focused on the ethanolamine utilization cluster. And this shows a data set where we actually analyzed anaerobic growth in a defined medium with ethanolamine as a control and with added B12 to see what the effect was of B12 addition. And that's the blue symbols. And what you see is that if you look at the colony forming units, there's a massive increase in the colony forming units if B12 is present. So defined medium ethanolamine requires B12 addition and to support growth. And if you look at ethanolamine metabolism, top right, then you see that ethanolamine is only metabolized when B12 is added. And if you look at the bottom, you see that the typical expected products of ethanolamine metabolism, acetate and ethanol, are indeed produced in these conditions and not the control conditions. And what we find is a ratio of about 2:1, whereas the prediction will be 1:1 based on redox balance. But I will come back to that shortly later. So we did proteomics, and what you actually see is that if you look for the EUT-induced cells, so they are again with B12, and you find massive upregulation of almost all proteins and that are, or genes that are in the cluster, and also translated into, let's say, very high protein levels. And when we combine this with transmission electron microscopy, what you see indeed in the control cells is that, and that the several microcompartment type as in line with how they are actually found in other microbes, and you can actually see these microcompartments appearing in these conditions. You may have noted that the shape and also the size is about 40 to 80 or 100 nanomolar, mimics bacteriophages. So it actually mimics bacteriophage heads, but there's no evolution constraint between that. So it's really microcompartments and protein shells and with the enzymes contained inside. So this is our final model for the utilization of ethanolamine by Listeria and via microcompartments, and this shows some additional information. And you see this sort of a threads that actually connect certain of the proteins to the shell proteins indeed. And there's still an ongoing discussion, how is this formed, how is this synthesized in time, and do we first have a sort of a shell already and then the enzymes are put in, or do we first have aggregates of enzymes and then they are combined with the shells? There's a lot of interesting studies going on that part. And what you get is ethanolamine goes in, B12 dependent conversion into aldehyde. What you also note is that ammonium is produced because ethanolamine has an amino group. And this also may play a role, and I won't go into that any further, but it could also play a role in pH control. So because of the release of ammonium, it might affect in close connection to the cell that actually the surroundings become more alkaline. So it also might link to maybe a bit of a sort of acid defense. So we find acetate and ethanol, but we find 2 acetate and 1 ethanol. That actually means that there are 2 ATP generated from 3 molecules of ethanolamine, whereas the prediction will be in a 1-to-1 ratio, you have 0.5 ATP per ethanolamine. So this is a 33% increase, which is of course massive. And we actually found out the mechanism, and if you're interested, you can read it in the paper, because this links to extracellular electron transfer connected with the microcompartment. So I think this is a very nice addition to the information contained to these kinds of metabolites and pathways. To connect it to the work of the Cornell group, and we actually produced filter sterilized cold smoked salmon food medium. So we have a sterile medium based on cold smoked salmon. And we actually looked for growth of Listeria monocytogenes at 7 degrees in anaerobic conditions. And we find indeed, that's the green and red lines, we find growth in this medium. But if we add ethanolamine, we see that the blue and purple line actually show massive increase in OD and also massive increase in coliform units. But here you see that there's very good growth with the addition of ethanolamine without additional supplementation of B12. And notably, B12 levels are very high in cold smoked salmon. So there's, of course, a couple of questions coming up. So is ethanolamine released in cold smoked salmon? And that's the first one. And how? And the other thing, the other point, of course, is what kind of enzymes or what kind of factors would allow Listeria to release it? And for this, actually, it brings into the topic that was discussed many times this morning, and so microbiota. And of course, you have co-microbiota in foods, and we also have co-microbiota in the GI tract. And if you look into the spoilage flora in cold smoked salmon, so vacuum-packed cold smoked salmon, and that's important, and storage at low temperature, there's a very typical spoilage flora, and that's mostly lactobacilli. And there's several lactobacilli that actually are lipolytic. And so, of course, we all know they are proteolytic, but they also are lipolytic. So we actually wonder, we're actually still working on that. And so how is actually ethanolamine released? Is the system activated in foods, or is it indeed sort of a cooperation, and that actually is derived from the spoilage microbiota? And of course, as always, further study is required. So we used a similar approach for the propane-diol cluster, and of course I won't show all the data, but we actually confirmed growth activation using all the techniques I mentioned. And also here, there's no growth in aerobic conditions, so a bit of oxygen already blocks this activation. And also here we find that addition of B12 is required. So we can find a situation where the B12 cluster in Listeria is activated and that it actually produces sufficient B12 to, let's say, give active microcompartments. So the question was, can BMC PDU, the next one, be activated and stimulate anaerobic growth with rhamnose? I already mentioned that rhamnose can produce, rhamnose metabolism can produce propane-diol. So actually, if propane-diol metabolism could be activated in this situation, maybe the propane-diol could be converted, let's say, to these other compounds and generate extra energy. So for this you have to know that Listeria is very well capable of growth on rhamnose. So this is, has been partly published before, but we checked the genome and also looked for the proteins. So actually our proteome analysis showed activation of the enzymes encoded by the LMO numbers and also by the Ra-M and Ra-A. And these enzymes are all activated during growth on rhamnose. So you can grow Listeria in aerobic conditions on rhamnose, but also in anaerobic conditions. But nobody ever checked what happens if you grow them anaerobically with addition of B12. So that's what we did. So this is actually how Listeria is growing aerobically or anaerobically with rhamnose. So it produces acetate, lactate, and 1,2-propane-diol. And we actually incubated Listeria in defined medium with rhamnose, without and with B12, with oxygen and without. And I highlight the anaerobic results because only in anaerobic conditions with the addition of B12, we indeed see that propane-diol levels are decreased and propionate and 1-propanol are produced. So this indicates that if you supply B12, Listeria can now convert propane-diol indeed to propionate and propanol. So we again checked microcompartment synthesis, etc. And our model now is shown here. So in the appropriate conditions, anaerobic conditions, with B12 added, we actually see a shift in metabolism of Listeria on rhamnose. So we have propane-diol reduced but still produced, and now we see an increase in propionate and propanol next to lactate and acetate. And this actually means that Listeria can enhance its efficiency on anaerobic growth by producing microcompartments if B12 is there. And as indicated, and the conversion to acetate, or sorry, to propionate, produces additional ATP, which is of course very important under energy limitation conditions and anaerobic conditions. And I will skip this part and move to the conclusions. And this was about the violence, and we actually did violence studies using transversal systems, where we could actually look for invasion, adhesion evasion, and also translocation of Listeria. And what I want to conclude is that, so what we actually saw was that upon the activation, and we actually checked all our results again, so if you then look at the proteomics, what we actually see is activation of stress defense. We find upregulation of CLIP chaperones, which are involved in protein quality. We find SOS damage repair proteins, and this all makes sense because, as you know, if you activate a pathway with a toxic intermediate, it has to be a very efficient fine-tuned system before it's optimally working. So on the way to success, there might be collateral damage, and that is what we see here. And we also find typical proteins upregulated that might be associated with the enhanced violence of these type of cells. So to conclude, BMC, UTE, and PDU support anaerobic growth with these compounds. Activation of BMC and PDU in Listeria requires addition of B12. We never could find a condition where its own synthesis was sufficient to support growth. And BMC-induced cells show activation of stress defense and enhanced in vitro violence. And based on the conditions and environmental niches, BMC, UTE, and PDU may enhance competitive fitness and anaerobic niches. And I indicated on the right, this is from an opinion paper that Andy published together with Alexander Dunk, another PhD student. And there's some very interesting aspects in the co-microbiota that may play a role in this whole process of this interaction, and also vitamin B12. So is vitamin B12 exchange required or not? And with this, I would like to go to the final slide. Thanking Annie again, was supported by Chinese scholarship and the co-supervisors of the thesis, Richard Notenbaert and Eddie Schmidt, and of course, MSc students and also cooperators that are on some of our papers. So thank you very much.
Thank you, Professor Abe, for very very much inspiration in these lectures, and this would need now at least a couple of hours of discussion. But unfortunately, your time is over, and we should be polite with our keynote speakers to give at least possibility for one question. Who is going to be the one? Everybody understood everything.
If nobody has a question, I have one.
Please.
Jacko, I'm a mere food microbiologist, but anaerobically, is there a relationship between the temperature and an anaerobic situation? Does Listeria grow better or worse at low temperatures? And can you do something? I mean, I'm thinking of vacuum-packed smoked salmon.
Well, like most microbes, it shows reduced growth rates at low temperature, but it can still grow very efficiently at low temperature, and also even in anaerobic conditions. So that's the exception is it's well tuned to also grow at low temperature.
Yes, but can you put it at sufficient without freezing it? Will it help if you have it at a very low temperature?
Yeah, it delays growth, so let's say the onset of reaching critical values or levels of Listeria is much delayed. And of course, you try to find other ways with preservatives, etc., to control it.
Yes.
Yeah? Okay. Thank
you once again.
Oh,
yeah, we have one more. Okay.
Yeah.
Great question. Kind of naive. Sorry. Fantastic talks, Jacko. I just had a kind of a naive question for you on these microcompartments. One thing is, can they be isolated? Could they work in vitro? And also, when you show those TEMs, you know, when we do TEMs of different organisms, we see them as well. But we see now what looked like what we thought were phage heads. And I like the analogy with the phage heads inside them. And so inside those TEMs that you saw, like, are they just the ethanolamine ones, or are they multiple different ones? So I had a few questions there.
You mean the numbers of the microcompartments? Or?
Yes, the microcompartments.
Yeah, what we see is it changes, maybe varies between 3 to 6, 8, something like that.
Can you speak to microphone, please?
Oh, sorry. So what we see, we see 3 to 6 or 8. So of course, if you have a phage burst, as you discussed this morning, you see massive loads of phage heads. But it looks very similar. So you have to do density centrifugation or isolate them, characterize them with the proteomics.
And will they work then if you feed them substrate?
If you isolate them, and actually, you can actually feed them substrate, just then, it has not been done with Listeria microcompartments, but the nanotechnology is actually aimed to produce them in vitro and actually use them for production.
Yeah, that sounds really exciting. Yeah. Yeah.
Thank you. And now, organizer would like to express our thanks to the professor, Professor Corey. Thank you. And thank you to everybody. Show goes on, and we leave the stage.